So these are some of the information available for the SIR2009 practicals for microbial ecology. So for the first practical, you will be tasked to make a 3 Winogradsky column. So this follows the contribution from Sir J. Winogradsky, the father of soil microbiology, when he used Winogradsky column to find microorganisms related to chemolithotrophy and other processes. Okay, so if you look here, we have 3 columns, one a control, and a B and C. Here we have cellulose, calcium sulfate, calcium carbonate, ammonium chloride, and dipotassium hydrogen phosphate. Okay, the only difference between B and C is in B we use calcium sulfate, whereas in C it is ferrum sulfate. So when you are making a Winogradsky column, you will be given soil, okay, where you are supposed to add tap water and create a slurry. A slurry is a semi-liquid kind of a consistency. We do it so that it doesn't trap any bubbles here, so that when we prepare the column, no bubbles are trapped, and then we overlay the soil column with water. Okay, so theoretically, when you have made a column that is done properly, you should be able to get a nice decreasing dissolved oxygen profile from the surface all the way to the soil area. Okay, so the dissolved oxygen profile will be from aerobic status to microaerophilic to anaerobic status. With time, after incubation in the under the sun, about 4 to 5 weeks later, you should be able to see different microorganisms developing at different layers of the column. So here are samples from previous experiments. This is the initial stage where you add the soil column with the water, and you can see that they are all the same. By week 2 already you can see some differences. There are greenish color in all the 3 columns, and the soil tends to become darker. Then by week 3, the differences between the columns begin to show. Here and here are essentially dark, whereas for the control column, the phototrophs have covered throughout the Winogradsky column. Another clear difference is the orangey tinge here, probably from the ferrous addition in this column. By week 4, you begin to see purplish tinge in column B, and here the orange tinge still remains around here. And finally, by week 5, the whole control column, lots of algae, cyanobacteria, except for the anaerobic area here. Whereas for this column B, you see whitish tinge. This is probably from calcium sulfide depositions, because in column B, calcium sulfate was added, but in column C, ferrous sulfate. Therefore, this one remains black because of the ferrous sulfide. This one becomes clear whitish because of the calcium sulfide. So under the microscope, some of the organisms you can see sheathed and filamentous microorganisms together with some phototrophs, photosynthetic microorganisms. So these are the questions for Winogradsky column. Why do we ensure that no bubbles are trapped? How do the physical chemical variables change throughout the column? Can you relate the changes in your Winogradsky column with the chemicals that you have added? Okay, so for the next practical, we will be repeating the Azotobacter isolation using nitrogen-free medium carried out by Martinez Bioring. This uses the approach of everything is everywhere, the environment selects. So this is the flask containing the N-free medium. This N-free medium has already been inoculated with surface soil. Then after 2 weeks, dilution streaking of the N-free broth reveals mucoid microorganisms growing. The plate used here are also nitrogen-free plates. So these are the questions. Do you think all the microbes growing in your enrichment culture are nitrogen fixers? Explain. Why do we not shake the flask or disturb the surface film? Everything is everywhere, the environment selects. Explain the statement within the context of this experiment.